The topic of my presentation is hydrogen peroxide and amyotrophic lateral sclerosis from biochemistry to pathophysiology. Free radicals are implicated in the pathogenesis of various neurodegenerative diseases. One of the complex, incurable, and life-threatening motor neurodegenerative disease is amyotrophic lateral sclerosis. The abnormal misfolding and aggregates of SOD1 and TDP43 is largely seen in the pathogenesis of both forms of ALS, that is sporadic form of ALS and familial forms of ALS. Several studies over the past 2 decades have shown that SOD1 catalyzed biochemical reaction product hydrogen peroxide act as a pathological concentration act as a substrate to trigger the misfolding trajectories toxicity of SOD1 in the pathogenesis of ALS. So hydrogen peroxide in the physiological concentration act as a oxidative U-stress molecule in cell signaling process, whereas in the pathological concentration it act as a oxidant and causes cell death. The toxicity of hydrogen peroxide is mainly due to the production of nature's most toxic radical called hydroxy radical due to Fenton's reaction and Haber-Wey's reaction. SOD1 is a homodimeric molecule and cysteine 111 act as a hotspot for oxidative modification of H2O2. The hypothetical model we proposed here shows how pathological concentration of H2O2 causes aggregation of both SOD1 and TDP43. If we follow the path A, we can see that pathological concentration of H2O2 causes sulfinylation, thereby oxidative modification of cysteine 111 here, and then causes demethylation. The demethylation process causes dissociation and hence aggregation of SOD1, which is toxic to motor neurons. If we follow the path B, we can see that pathological concentration of H2O2 causes post-translation modification by sulfinylation and demethylation process. This sulfinylation process could cause disruption in the TDP43 nucleocytoplasmic shuttle, and thereby the TDP43, instead of going to the nucleus, it aggregates outside in the cytoplasm and form the oligomeric neurotoxic aggregates. Both the SOD1 and TDP43 aggregates are neurotoxic and causes degeneration of motor neurons in ALS. Literature from the past 20 years has shown that changes in the redox property of SOD1 causes abnormal conformational changes in the structure of SOD1 protein, leading to the gain of toxic properties, destroying motor neurons in both forms of ALS, that is sporadic and familial ALS. Higher concentration of H2O2, that is called pathological concentration of H2O2, act as a determinant or precursor to generate nature's most reactive, harmful and toxic species called hydroxyl radical, thus changing the cellular redox thiol status of SOD1 and TDP43 proteins. So thereby we highlighted the role of hydrogen peroxide in ALS. We believe this would encourage scientists to target pathological concentration of H2O2, thereby halting the misfolding of SOD1 and both TDP43, which is implicated in largely in the pathogenesis of ALS. Thank you.